Enzymes play an extremely important role in our body by helping to speed up and control the rate of chemical reactions within our body. Now, of course, in the last lecture, we looked at some of the mechanisms that enzymes use to speed up our reaction rates. In this lecture, I want to look at how much our enzyme increases our reaction rates. So we're going to take a quantitative look at enzyme kinetics. So kinetics is the study of the rate at which compounds react. The rate of our enzymatic reactions will be affected by what enzyme we're using, so our catalyst, the amount of substrate, the substrate concentration, any different effectors, as in allosteric regulators or drug molecules, things like that, and then, of course, the temperature as well. There are a couple different reasons why it's important to understand our enzyme kinetics. The first one is that studying our kinetics can give us insight into the mechanism and behavior of the enzyme, right? So it's often the first step to allow us to potentially intervene and change the rate of an enzyme. If we know how it works, then we can know something about how we might be able to fix it if it's broken. So that understanding of our kinetic mechanism is closely tied to regulation of that enzyme. And again, it can often provide us insight into how we can change the enzyme's activity. In order to quantitatively describe our kinetics, it's important that we have a model for our kinetic mechanism. So we're going to first start with the model mechanism. Then we're going to identify our key constraints and assumptions for that particular model. And then we'll be using some algebra in order to develop an equation that we can use for our calculations. So at its heart, the simplest possible model we can use for an enzyme is shown here, right? We have our enzyme marked as E, binding to our substrate S that forms our enzyme-substrate complex, which then reacts to form our free enzyme and our product. So of course, this model that we talked about in our last lecture when we talked about different enzyme mechanisms. And in this case, of course, we have one reactant, one product, and no inhibitors here. A lot of enzymes, of course, will be much more complex than this. We might have multiple reactants, or we might have inhibitors present, but we'll deal with those complications later. Now that we have our basic model, we can identify the key constraints and assumptions that we're going to need to use in order to quantitatively describe this. So, first off, we're going to assume that the total enzyme concentration is constant. So this is a pretty easy assumption, right? Enzymes, like all catalysts, aren't consumed by the reaction, so we won't be losing our enzyme over time. So we'll have our total enzyme concentration will always be equal to the free enzyme plus the enzyme substrate complex concentration. We also can usually assume that our total substrate concentration, which of course would be equal to the free substrate concentration plus the substrate bound in our enzyme, is roughly proportional to the free substrate concentration. Again, this is an assumption that we used before when we were looking at ligands binding to proteins. So we usually assume that the substrate has a much higher concentration than our protein. And so the substrate concentration isn't significantly decreased by binding to our protein. So the observed rate of our reaction would be based on the change in product over a certain time period, a change in time. So we call this our reaction rate over here with our little V here representing that rate. And this is proportional to our rate constant times the concentration of our enzyme substrate complex, right? Our rate of product formation is based on just this second key step here. So the rate of product formation is also the rate of breakdown of our enzyme substrate complex to form that product. Now, the final key assumption that we're going to make here is what we call the steady state assumption. So what we're going to assume is that after our enzyme gets going, we're going to get to a point where the rate of formation of our enzyme substrate complex is equal to the rate of breakdown of the enzyme substrate complex, right? So we're going to have substrate binding in, and then we're going to have product coming off of our enzyme at about the same rates. So our concentration of the enzyme substrate complex actually won't change once kind of our enzyme gets going and is in its full flow of producing our product. So if we go back to our model here, we can now apply rate constants in here for each of these processes. So we'll have a rate constant for the formation of our enzyme substrate complex, a rate product, an inverse rate product for the breakdown of that complex back to our free enzyme and substrate, and then a second rate constant for the actual reaction that produces our product inside of the enzyme. So now with these rate constants, we can go back to our steady state assumption, and we know that the rate of formation of our enzyme substrate complex will be based on our rate constant multiplied by the concentrations of our involved components. That would be the concentration of the free enzyme and the concentration of our substrate. So that's the rate of formation of the enzyme substrate complex. The rate of breakdown of the enzyme substrate complex is based on the rate that our product forms and it's also based on the rate that the enzyme substrate complex breaks down. So this would be K minus 1 times the concentration of the enzyme substrate complex. So each of these rates should be equalized based on our steady state assumption. Now, one of the issues that we have with our kinetic measurements is that we can't necessarily easily measure all of these different concentrations. Usually we'll have a very clear idea of what our substrate concentration is. That's often something that we can fairly easily measure, or maybe we can control exactly how much substrate we add. We usually have a good idea of what our total enzyme concentration is. Again, maybe we've added a certain amount of enzyme in order to start up this experiment. But it's not necessarily easy to know exactly how much of our free enzyme we have or how much of our enzyme substrate complex we have. So we can use these rates in order to develop an expression for the concentration of our enzyme substrate complex. So we can use our known concentrations in order to try to calculate that enzyme substrate complex concentration. And that can then be applied to our equation here to determine what our rate will be. So I'm not going to show the full derivation of this, but using the steady state assumption together with our known concentrations here, we can develop an expression for our enzyme substrate complex and substitute that in to create the Michaelis-Menten equation. So here's our Michaelis-Menten equation here. So notice we have our rate over here on the left. That's the rate of our reaction. We have the substrate concentration. Again, this is usually something that we will know based on how we set up the experiment. And we have two other key constants here. So our Km here is the Michaelis constant. So this is a combination of some of our different rates from that simple enzyme reaction model there. So it's actually equal to K minus 1 plus K2 over K1. So essentially the rate constants for the formation, or the rate constants for the breakdown rather, of our enzyme substrate complex divided by the rate constant for the formation of our enzyme substrate complex. So this is based on our simple model. If you have more complex models of an enzyme, which again we won't deal with in this course, the Km, you would still use the Km, but it might have a more complex meaning in terms of what rate constants are hidden there in that Km. So our Km is just a combination of those different rate constants. Our Vmax up here is the maximum possible rate of our reaction. Now, this would occur when all of our enzyme has substrate bound. So our entire enzyme concentration is equal to our enzyme substrate complex concentration, or in other words, the total, total concentration of our enzyme is equal to our enzyme substrate complex. And then, of course, the rate there would be based on our rate constant for that breakdown. So this would be essentially K2 times E total is equal to our Vmax. K2, again, being the rate constant, specifically dealing with the conversion of our substrate into our product in our enzyme complex. So at this point, then, we can rearrange our Michaelis-Menten equation, and we can also express it like this. So K-cat here is that K2, essentially, saying how quickly our complex reacts to produce our product. We have our total enzyme concentration, and this, of course, again, just simplifies to our Vmax. So it's just showing what composes our overall Vmax constant here. The K-cat is also called the turnover number. So it tells us how many substrate molecules one enzyme molecule can convert per second, right? How much of our substrate is turning into that product for that particular enzyme. The exact meaning of our Km and Kcat, again, will actually depend on the details of our mechanism. Kcat equals K2 for that simple mechanism that we looked at, but as you go to more complex mechanisms, you might have other rate constants involved in that as well. Same with our Km there before, where again it was K minus 1 plus K2 over K1. So again, both our Km and our Vmax and our Kcat are all constants for a given enzyme. The Vmax can be modified by changing our total enzyme concentration. So it's not constant at any concentration of our enzyme, whereas our Kcat and Km are more just based on what the enzyme mechanism, how it is, and how that enzyme works. So Km and Kcat won't directly change for a given enzyme, regardless of what concentration it is. So the Kcat and Km are constants for a given enzyme and can be used to indicate the overall efficiency of our enzyme. So we call this, if we divide our Kcat by the Km, this gives us a specificity term. So that the Kcat-Km ratio refer to as the specificity of a particular enzyme. This specificity will max out around 10 to the 8th or 10 to the 9th. This is just based on the limits of diffusion, right? So molecules have to be able to move or diffuse into our enzyme and then diffuse out as the product before a new molecule can come in. A lot of our enzymes actually do get close to this theoretical maximum limit. So if you look at these specificities down here in our table, you'll notice that a lot of these are very close to they're around the 10 to the 8th range or 10 to the 7th range, close to that kind of maximum limit. So notice how quickly and how efficiently most of our biological enzymes work. Now, of course, since this is a ratio term, our overall enzyme efficiency can be controlled by changing our Kcat or our Km, right? So we would say that you can increase the velocity, right? Increase that turnover number, the rate at which we're reacting inside our enzyme, or you can also increase the affinity for our substrate. This affinity is kind of roughly correlated to our Km. So the Km is kind of a measure of how well our substrate binds to our enzyme, although that's a little bit of an oversimplification of the Km. You might notice in our actual Michaelis-Menten equation, it actually looks very similar to the equation for a ligand binding to a protein. So remember, in our ligand binding equation, we had the total fraction of ligand bound to our protein was equal to the ligand concentration which again corresponds to our substrate concentration here, divided by the Kd plus the ligand concentration. Again, notice our ligand concentration, of course, corresponds to our substrate concentration for our enzyme. And so our Kd actually kind of takes a similar role to our Km in the Michaelis-Menten equation. So again, kind of showing that Km kind of does roughly indicate the Kd of our enzyme, although, again, it is more complicated than that. We do have other rate constants that are rolled into that Km as well. You might remember that our Kd expression actually relates to the rate constant for the dissociation from our enzyme substrate complex, or protein ligand complex in this case, over the rate constant for the association, right? My original definition of the Km was K minus 1 plus K2 over K1. So Km actually equals the Kd if you have very small values for the K2. So how do we actually measure these key constants of our Km or our Vmax and our Kcat? Well, to do this, we would use a kinetic experiment where we combine our enzyme and substrate together. We would use known concentrations of each, of course. So that would give us our E-tot, our total enzyme concentration, and of course, our total substrate concentration, which we usually assume is roughly equivalent to our free substrate concentration. And then we would record the rate of either substrate disappearance or product formation. Usually we might try to do this by measuring the absorbance of the substrate or the product and relating that to the concentration. But it would depend on, of course, what substrate and product you're forming here. We can then plot the initial velocity versus our substrate concentration, and then just change our substrate concentration and repeat. So our experiment might look something like this. So we have our absorbance, which is telling us, in this case, the substrate concentration. So we mix our reaction together, and then we have this initial velocity determined by the slope as our substrate is consumed. We would only want to use this initial period because after that initial period, our reaction is going to slow down as our substrate is consumed, and our assumptions will no longer apply. If we express this graph again in terms of product concentration, so what we'll see is our product concentration will increase at essentially a linear rate initially, and then it will slow down as we use up our substrate and our reaction rate changes. As we change our substrate concentration, our rate, of course, will also change. So we'll want to measure that initial rate at multiple different substrate concentrations so that we can then get an idea of our rate at different concentrations here. Once we've measured our initial velocity at a variety of different substrate concentrations, we can make a graph comparing our initial velocity with our substrate concentration. Now, of course, the Michaelis-Menten equation tells us that our rate, in this case our initial velocity, corresponds to our Vmax times the substrate concentration over our Km plus the substrate concentration. So our Vmax, of course, is the maximum rate at which our enzyme proceeds. So our rate will slowly approach that Vmax as we increase our substrate concentration, but it will never quite reach it. So the Vmax will be the point of our initial velocity that's kind of just above where our velocity levels off as we increase our substrate concentration. The Km here actually has a similar definition to, again, the Kd that we saw previously with our protein ligand binding interactions. So if we are at half of our maximum velocity, our substrate concentration will be equal to our Km. And of course, we can quickly prove this by substituting this into our equation, right? So if we say our Km is equal to our substrate concentration, the bottom of this equation simplifies to 2s. Our substrate concentrations will then cancel out, and our rate is equal to Vmax divided by 2. So our graph of velocity versus substrate concentration provides us with a simple way of trying to measure our Km and our Vmax. Again, the Km would be that point where we reach half of our maximum velocity, and our maximum velocity is the point at which we saturate in terms of our substrate concentration, and we don't see our rate increasing anymore as we increase our substrate concentration. Now, it's not always easy to estimate these from our graph, so it's helpful to have a more quantitative method of measuring these key kinetic parameters. So there are a couple different ways we could do this. First, we could convert our graph into a linearized double reciprocal plot. So in order to do this, we need to convert our graph, right, which was obviously nonlinear, into a straight line. Now, again, the equation for a straight line would be y equals mx plus b. Our current equation is rate, our initial rate v0 equals vmax times the substrate over Km plus the substrate. If we take a double reciprocal of this, that means inverting both sides of this equation. So we would go 1 over our initial rate equals Km over Vmax over the substrate plus the substrate over Vmax times the substrate. Now notice our substrate concentrations will cancel out over here, so this just becomes 1 over Vmax, Km over Vmax, 1 over S. And if we look at our original equation for a line, you'll notice we've actually turned this into a linear equation. So Km over Vmax becomes our slope, our y value becomes 1 over our rate, our x values become 1 over our substrate concentrations, and our intercept will be 1 over Vmax. So this is a very simple way of measuring our Km and Vmax, and we'll look at that in more detail in just a minute. But I do want to mention there are a couple of other methods we can use as well. We could use a nonlinear Michaelis-Menten plot, and we could then use a nonlinear fitting method to calculate the parameters directly from that graph. So this tends to be more accurate than our double reciprocal plot, although it's a little bit harder to do because you need a computer program to help with that nonlinear fitting. Our linearized double reciprocal plot is also helpful because it can be used for analysis of some of our other more complicated enzyme mechanisms as well. So when we add in multiple substrates or when we have inhibitors present. So let's take a closer look at our double reciprocal plot. And this is also called a Lineweaver-Burk plot. So again, as I mentioned, we have our y equals mx plus b linear form for our equation, where our slope, our m, is Km over Vmax. Our intercept is 1 over Vmax. And of course, our y-axis is 1 over the initial rates. And our x-axis is 1 over the substrate concentrations. So you can immediately notice that it's very easy here to calculate our Vmax and our Km. Right? You could easily plot a linear line on here, and you just need to see where it hits the y-axis to get your Vmax, and then you can measure the slope and use the Vmax to calculate the Km. You can also get the Km from the x-intercept, so where our line hits the x-axis will be negative 1 over Km. So you could also get the Km from there if you didn't want to calculate the slope. A second method of linearizing our Michaelis-Menten plot is the Adey-Hofstee plot. So in this method, we, instead of using our double reciprocal approach, we include our rate in both the y and the x value. So our y value is the rate, and our x value is the ratio of the rate over the substrate concentration. You can see in this case, our slope would be negative km, and our y-intercept would be the Vmax instead of the reciprocal of the Vmax. Both of these plots are very helpful for measuring those kms and Vmax quickly, but they both do introduce some errors due to their uses of reciprocals and this ratio here within our x value here. So far, we've only looked at relatively simple reactions with a single substrate combining into our enzyme-substrate complex and then reacting to form our product. Again, many of our reactions will involve more than one substrate. So I want to briefly show you how our model is modified if we have more than one substrate. So first, we need to understand what the kinetic mechanism of our enzyme will be. So this will depend on the order of binding of our different substrates and the release of our products. In general, there are a couple of different mechanisms that we can use. We can either have a sequential mechanism, where our substrates bind in a certain order and then are released in a specific order. So for this mechanism, our key is that we have a complex with multiple substrates in it at the same time. So they'll all bind together and we'll get a multi-substrate complex at some point, and then they'll all be released to get our products. Our other main possibility is a ping-pong mechanism. So in this case, we have a transient modification of the enzyme. So we'll have one substrate that will initially bind to our enzyme and then modify our enzyme, and then it will be released, and we'll have our second substrate that will come in and interact with that modified enzyme before restoring our enzyme to its original structure. Let's take a look at the model systems for each of these different approaches. So first we have our sequential approach. Again, this would involve a ternary complex where we have two substrates along with our enzyme together. So we can either have an ordered model where we have one substrate bind and then the second substrate bind in a specific order before releasing those two products. Or we can have a random order where either substrate can bind first, and then we can reach our enzyme-substrate ternary complex. In order to represent these models more quickly and efficiently, we can use this Clellan nomenclature over here on the right. So in this nomenclature, we have one line that's kind of our reaction coordinate. So we have our enzyme marked here as E on the bottom. Our initial substrate comes in as A, so we see that represented coming in with an arrow, and we form our enzyme-substrate complex. Then our second substrate is marked after A alphabetically as B. We get our EAB complex. This then reacts to create our product, P, and then our second substrate becomes the second product, Q. Q, of course, used because it's directly after P in the alphabet. And then we show our two steps of our product being released. So this would be an ordered by mechanism, meaning that we have a specific order for the two substrates coming in, two referring to the by prefix there, and then a specific order for our two products leaving. We can also have a random by down here. So we have our enzyme, and then we have two possible paths where either A could bind first and then B, or B could bind first and then A. They both result in our ternary complex. We then have our reaction, and then a random order for which product is released. So again, it's just a quick way of showing what order our products are coming in, what order they are leaving. Our ping-pong mechanism is shown down here at the bottom. So again, in the ping-pong mechanism, only one substrate is binding at a time, but it's modifying our enzyme. So substrate 1 binds, it reacts, and now we have our modified enzyme represented as E prime with our little apostrophe here. So that's just indicating that our substrate was modified, or has modified our enzyme in some way as it produced our first product. That product is released, and then our second substrate comes in, binds that modified enzyme, produces our original enzyme and our second product. We would represent this in Cleland nomenclature in a similar way to our sequential mechanisms. So we have the first substrate coming in. Now, when we react our enzyme, we use the next letter in the alphabet, F, to represent our modified enzyme. So we get FP. P is released to produce our F, our modified enzyme. B comes in and binds to our enzyme as our second substrate. That reacts and restores our enzyme and produces our second product to complete our mechanism here. Now, of course, as you can imagine, there are also many other possibilities for what these ping-pong and sequential mechanisms might look like. But we can all represent those using this kind of basic Cleland nomenclature. Now, if we were to measure the rates based on the change in substrate concentration for a sequential kinetic mechanism, we'd actually be able to easily distinguish this based on the shape of our Lineweaver-Burk graphs. So this is one of the advantages of that Lineweaver-Burk graph, is it actually tells us something about our mechanism even beyond just that Km and Vmax value. So what you do is you'd make a graph based on the initial rates versus one of your substrate concentrations, and then you'd slowly change the other substrate concentration and keep measuring the same kind of graph again. So you'd measure a series of different substrate concentrations for substrate 1 along with their rates, change substrate concentration 2, and repeat it again until you get several different lines for your Lineweaver-Burk plots. With a sequential mechanism, you'll always get your lines intersecting. So if you see your lines intersecting, that indicates a sequential mechanism. You also can determine whether it's random or ordered based on where the intersection point occurs. So for a random mechanism, your intersection point will occur at the y-axis. So all of our lines would be intercepting right here. That would represent that your Vmax value essentially stays the same regardless of how you change your substrate concentrations. For a ping-pong mechanism, we'll see a key difference in our Lineweaver-Burk plot. Our lines will actually be parallel here. So in this case, the slope will be constant between each line as we change our substrate concentration, and we'll just change where we intercept the y-axis. So again, as long as we can measure the rate at different concentrations of substrate 1 and substrate 2, we could actually use this to learn something about the mechanism of our 2-substrate enzyme. So we can already see from this that these kinetic experiments are very useful for helping us to know how our enzyme works. Now, in the next lecture, we'll see that these sorts of enzyme kinetic measurements can also help us to know something about how our enzyme is regulated by either other molecules within the body or different drug molecules or inhibitors that we might add.